Hello, everyone. My name is Michelle Lee, and I'm a Berkowitz Postdoctoral Research Fellow at Harvard Medical School. In this lecture, I will be covering the key advancements in artificial intelligence and machine learning on graphs, and showcasing their impact on accelerating research in precision medicine. If you haven't already, please be sure to first watch the other Day 1 lectures, particularly the Principles of Network Science lecture by Dr. Kimberly Glass. Let's get started. Unsurprisingly, biology is interconnected. Our proteins interact, so they form a network where the nodes are proteins and edges represent some form of interaction between them. Non-coding RNA interact with each other as well as with proteins. Drugs target proteins and treat diseases. Drugs can interact with each other to enhance efficacy or lead to adverse events, and lots more. But so what? Why does this matter? This interconnectedness we see in biology is important because the effects of drugs, for example, are not limited to the molecules to which they directly bind in the body. Instead, these effects spread throughout the biological networks in which they act. So the effect of a drug on a disease is inherently a network phenomenon. Networks have been used as a general language for describing and modeling complex systems like biology and medicine. With the help of networks, we have been able to discover new biology. For instance, using patient networks to identify new disease subtypes with distinct patient survival, simulating a basic eukaryotic cell to reveal mechanisms of cell growth, drug resistance, and synthetic life, and leveraging cross-domain biomedical knowledge to better understand the effects of drugs and mechanisms of various diseases. Biological networks come with their own set of challenges when it comes to machine learning. First, networks can involve heterogeneous interactions that span from molecules to whole populations. The challenge is computationally operationalizing these data and making them amenable to machine learning. For example, how can we model proteins, drugs, and diseases such that we can disentangle disease-protein interactions from drug-protein interactions? Secondly, networks need data from diverse sources, including experimental readouts, curated annotations, and metadata. Unfortunately, no single data type can capture all the factors necessary to understand a phenomenon such as a disease. So the question is, how do we take all of these data sources into consideration for our prediction task at hand? Third, networks are noisy due to inherent natural variations and limitations of measurement platforms. This results in missing data, repeated measurements, and contradictory observations, which can plague our analysis. Another reason why biomedical networks are challenging is that classic deep learning methods are designed for data with a regular grid-like structure, such as images which have a 2D grid structure and sequences which have a linear 1D structure. These models have brought extraordinary gains in computer vision, natural language processing, speech, and robotics. However, they are unable to truly exploit complex irregular interactions or edges between entities, which are the essence of graphs. Concretely, networks are far more complex because they can be of arbitrary size, have complex topological structure, have no fixed node ordering or reference point, and are often dynamic and have multimodal features. So we need deep learning methods that can handle graphs specifically. It would take as input a biomedical network and transform it such that we can leverage interesting structures in the graph to generate predictions. Ideally, we would see clustering of nodes based both on their features, represented by their colors, and structure. So the proximity of the embeddings approximately reflects the similarity and proximity of the nodes in the network. As an example, many studies have found that cellular components associated with a specific disease tend to cluster in the same network neighborhood. In the rest of this talk, I will try to convince you that deep graph learning methods are well suited for the analysis of biological networks. So let's start from the basics. What are the main graph machine learning tasks? We have node level classification, where we predict the characteristics of a given node. In this example, we have a protein interaction network where nodes are proteins and edges indicate physical interactions between pairs of nodes. With this protein interaction network, we can predict for each node or protein what function it has. Next is edge level prediction, where we predict whether or how two nodes are connected. In this example, given the heterogeneous network where nodes are drugs and diseases and edges indicate that the drug treats the disease, we can learn which diseases a molecule might treat by predicting whether such an edge exists between a pair of drug and disease nodes. We also have subgraph level classification, where we predict how clusters of nodes interact with each other and the rest of the graph. In this example, the goal is to better understand a disease's underlying mechanism. So we have the protein interaction network again, and this time we want to identify which set of proteins are involved in the disease. The hypothesis is that by extracting not only the proteins involved in the disease, but also the relevant interactions or edges, we can better disentangle the pathways that may be relevant for the disease. Lastly, graph level classification, where we predict characteristics of whole graphs based on similarity to other graphs. In this example, we want to design a new small molecule compound to treat a disease. Here, the graph is the molecule itself, where nodes are amino acids and edges represent bonds between amino acids. So, given a graph of the molecule, we can predict many kinds of characteristics about it, like solubility, binding affinity, etc. At the same time, we can even generate new graphs or new molecules with the desired properties to treat disease. To perform these four kinds of tasks, the core idea is the following. We automatically learn embeddings or low-dimensional feature representations for the entities in our graphs. For example, here we have a node embedding or a feature representation of a node in the graph. We can also have subgraph embeddings and graph embeddings. The intuition here is to map nodes in the graph to embeddings in a mathematical space such that similar nodes in the graph are embedded nearby. Now the question is, how do we learn this mapping function? There is a wide spectrum of predominant graph learning paradigms. Today, I will only briefly touch on the most commonly adopted one, graph neural networks. Graph neural networks aim to learn node embeddings by synthesizing graph structure and features via multiple layers of nonlinear transformations. Currently, most graph neural network models have a somewhat universal architecture. You may have heard of them being used interchangeably with graph convolutional networks. But before we go into what graph convolutional networks are, let's start with regular convolutional networks. Convolutional networks are typically used on images. The idea is that you use filters to learn its features. For example, in a single convolutional network with a 3x3 filter, you are aggregating information in that 3x3 region of the image into a single value saved in the top left corner of the green matrix. This is often done by taking an average. Then you will repeat with the next 3x3 region until you fill up the 5x5 green matrix. This idea of transforming information or messages from neighbors and then combining them is fundamental to convolutional networks. And so, of course, they apply to graph convolutional networks too. But what if your graphs look like this? Real-world graphs are not grid-like. As mentioned before, graphs are of arbitrary size, have complex topological structure, no fixed node ordering or reference point, often dynamic, and have multimodal features. So how do we address these issues? A naive approach is to concatenate the adjacency matrix with the feature matrix and feed it into a deep neural network. But this gives us O of n parameters, where n is the number of nodes. It's not applicable to graphs of different sizes and not invariant to node ordering. This brings us to graph neural networks. Again, the goal is to learn an embedding of any node in the graph such that the embedding captures information about the node's local neighborhood. To learn an embedding of node U in dark blue, a GNN propagates information along the edges through the nodes that are within k hops from U. In other words, we define each node's neighborhood as a computational graph and then generate node embeddings based on these local network neighborhoods. In this example, we have a target node A that we want to learn the embedding of. Node A has three neighbors, B, C, and D. B has neighbors A and C has neighbors A, B, E, and F, and D has one neighbor, A. These boxes are the neural networks. And there are three layers, where layer 0 is the node features X, layer 1 is the next layer that aggregates feature information to target A's one-hop neighbors, layer 2 is the final layer that aggregates the aggregated information from layer 1 into our target node A. These layers can be of arbitrary depth too. To aggregate information, a basic approach is simply taking the average of the neighbor's embeddings or feature information. There are, of course, more advanced ways to do this, such as using attention mechanisms, which allow you to automatically learn which neighbor's information should be weighted more or less than others. Here's what this looks like mathematically. H0 is the 0th layer, aka the node features. Hk is the kth layer. The red box is highlighting taking the average of the neighbor's embeddings from the previous layer. Hk minus 1 is the embeddings of the previous layer. Sigma is a nonlinear activation function such as ReLU. And finally, ZV is the embedding after k layers of neighborhood aggregation. We can feed these resulting embeddings into any loss function and then run stochastic gradient descent to learn the weight parameters in blue. Let's now refine our understanding of graph neural networks through two real-world applications in precision medicine. In the next part of this talk, I will briefly describe how we can integrate graph machine learning methods to generate predictions on context from the cellular scale to the patient scale. Specifically, through Pinnacle, we can investigate the roles of a protein across multiple cell type contexts and nominate therapeutic targets in a cell type specific manner. And through Shepherd, we can identify causal genes of a patient's rare or novel disease presentation and characterize never before seen disease presentations. Let's start with Pinnacle. First of all, context is important for understanding the behavior of genes and proteins. H2A of X, for example, is a gene that has many functions depending on its cell context. It is known that gene expression and protein function depend on cellular and tissue context, and biological functions can vary considerably across healthy and disease states. As such, therapeutic effects like adverse drug events due to off-target effects can vary based on context. So models that aim to generate predictions on proteins should consider the different types of contexts that they may be operating under, right? However, current protein representation learning approaches, including those that are structure and/or sequence-based, are context-free, as in each protein has only one representation learned from a single context or an integrated view across many contexts. And these context-free methods are unable to adjust outputs based on the context in which they operate. And so the question is, how do we develop models that can adjust their outputs to the biological context in which they operate? Here we introduce Pinnacle, a contextual GNN model that can enable precise protein representation learning and cell type or cell state specific prediction. It takes as input cell type specific protein interaction networks, which we constructed from a multi-organ single cell transcriptomic atlas of humans, and a metagraph of cellular interactions and tissue hierarchy. Given these networks, Pinnacle learns tailored protein representations depending on the desired cell type context. How does Pinnacle do this? First, let me give you some intuition for the model. Pinnacle embeds the protein, cell type, and tissue nodes in a single unified embedding space such that protein embeddings from the same cell type network are attracted to each other if those proteins share an edge, as in they're closer together in the embedding space. If they don't share an edge, then their embeddings are repelled from each other, as in they're farther away in the embedding space. Protein embeddings from the same cell type network are attracted to the corresponding cell type embedding and repelled from other cell type embeddings. Cell type and tissue embeddings are embedded nearby if they share an edge in the metagraph, otherwise they are repelled. The goal here is to learn an embedding space that is organized according to the behavior of proteins at each cell type based on network topology, interactions between cell types, and hierarchical organization of tissues. Let's go a bit deeper into how this is mathematically formalized. There are three key components of Pinnacle: protein level attention mechanism with cell type specificity, metagraph level attention mechanism on cellular interactions and tissue hierarchy, and an attention bridge between protein and cell type embeddings. First up is the protein level attention with cell type specificity. The superscript PP means that we're dealing with protein-protein edges. For each node, we aggregate the neighboring node's embeddings H using an attention mechanism alpha and a protein-protein edge type specific transformation matrix W to project the features of the protein in its cell type specific PPI network. There is one W for each cell type specific PPI network. This similar message passing is used on the cell type and tissue nodes in the metagraph as well, but separated by edge type, namely cell type to cell type edges, cell type to tissue edges, tissue to cell type edges, and tissue to tissue edges. Then we aggregate these different views of a node's neighborhood by learning an attention over the edge types. Finally, you may have noticed that there are no edges between the proteins and the cell types. To allow messages to be propagated between proteins and cell types, we learn an attention bridge called gamma. This means that to learn a cell type embedding, we take the attention weighted average of the proteins that are activated in the cell type. The same gamma is used to regularize the cell type specific protein embeddings. As for our objective function, we have three terms: loss at the protein level, cell type level, and tissue level. At the protein level, we perform link prediction on the protein-protein interactions. We also predict which cell type the protein is activated in. At the cell type and tissue levels, we perform link prediction only, separately considering the different edge types. For link prediction, we use binary cross entropy. For cell type classification, we use central loss. As a result, we generate a single unified embedding space where protein representations are organized according to the cell type context and tissue organization. In these UMAP plots, notice that proteins from the same cell type context are enriched in a particular embedding region. Separately from the UMAP, we use an approach called SAFE, spatial analysis of functional enrichment to show that indeed the spatial regions in Pinnacle's embedding space are significantly enriched for protein representations originating from the same cell type context. Altogether, Pinnacle provides contextualized protein representations, cell type representations, and tissue representations, which we can use to finally generate contextualized predictions with cell type resolution. Given our contextualized protein representations, we can fine-tune or adapt them for any downstream task of interest. In this case, we are interested in predicting whether a given protein is a strong candidate for a therapeutic area of interest. This is actually a binary classification task. Given a protein, is it a strong candidate? Close to 1 means yes it is, close to 0 means no. But because we have a unique protein representation per cell type context, we can additionally gain insights about which cell types are highly predictive to examine therapeutic targets. So how do we define protein targets? First, we have a universe of druggable proteins in our protein-protein interaction networks. These are proteins that are nominal targets of existing drugs. A subset of those proteins are proteins that have been studied in our therapeutic area. A subset of those have been investigated in clinical trials. A subset of those have been investigated in clinical trials that have at least completed phase 2 of clinical trials. This becomes our positive chaining examples, as in they have label of 1. This subset of druggable proteins that have not been studied in our therapeutic area of interest becomes our negative chaining example, as in they have a label of 0. The two therapeutic areas that we choose to study are rheumatoid arthritis and inflammatory bowel disease. On the x-axis is our performance metric, and the y-axis is the different models that we evaluate against. The first row is Pinnacle. Each dot here represents a cell type context. Notice how there is a spectrum in the performance of these cell type contexts. This suggests that there are some cell type contexts that are better at predicting therapeutic targets for RA and IBD than others, which is consistent with what we would expect. These next 3 rows are baseline models, all of which are context-free. For these models, we only see one data point shown by the square because these approaches cannot adjust their outputs depending on the context. Notice that for the RA and IBD models, there are many cell type contexts that outperform the baseline model. This means that Pinnacle's representations predict drug targets for RA and IBD significantly better in those cell type contexts than context-free methods. Let's take a look at what cell types they are and why there are some cell types that do not perform better than the baseline models. Here is a zoomed-in version of the performance for RA, except now the performance is on the y-axis and the cell types are on the x-axis. The dotted lines represent the baseline model's performance. Again, each dot represents the performance of proteins from a single cell type. We have validated with an RA clinician that the top-ranking cell types are highly relevant to RA and align with the literature, and the low-ranking cell types are not so relevant to RA. We can also look into individual proteins and examine the most and least predictive cell types to better understand the mechanism of action. We can see similarly exciting results for IBD, and we have worked with a clinician at Massachusetts General Hospital to verify our findings. I think these are really exciting times for single-cell biology and AI. While I agree with this article's position that we aren't quite at a point where single-cell analysis can be integrated into routine clinical practice, I do think that Pinnacle models bring us one step closer. That is, if we define precision medicine as being able to accurately match an individual patient to the most beneficial therapy based on the patient's genomic and molecular profiles, contextual graph machine learning models have the potential to enable exactly that, adjusting the predictions given a patient's context. I even believe that contextual graph machine learning broadly can realize the promise of precision medicine. Another aspect of my research is actually on innovating contextual graph machine learning models for rare disease diagnosis by leveraging patient-specific context. And this brings me to the next real-world application, Shepherd. To give you a little bit of background, rare diseases affect 1 in 20 people worldwide, yet each disease affects no more than 50 per 100,000 individuals. Rare disease diagnosis is challenging due to the considerable heterogeneity of clinical presentations and the large number of candidates that must be considered to identify molecular diagnosis. Many patients suffering from rare diseases are undiagnosed, and it takes 4 to 5 years on average for patients to receive a diagnosis. So our key question for this work is, could we leverage deep learning methods to accelerate the diagnosis of rare disease patients? We've seen deep learning methods achieve tremendous promise in other diagnostic areas. So it's a natural question to then ask whether we can apply these approaches to rare disease diagnosis. The challenge is that these advances crucially depend on the existence of large annotated datasets containing thousands of examples per condition for training deep learning models, whereas datasets for rare diseases are 3 orders of magnitude smaller. AI-assisted diagnosis of rare diseases encounters challenges that are distinct from those other use cases I just showed you. Given the lack of large annotated datasets, the approaches must be able to learn from limited data. Approaches must also be able to extrapolate beyond the training data to atypical disease presentations or never before seen genetic conditions. To give you a better idea of how challenging this is, let's take a look at real patients from the Undiagnosed Diseases Network. The UDN is a multi-site program in the US aimed to diagnose patients with rare diseases. Of 465 diagnosed patients in the UDN, there are 378 unique causal genes and 299 unique diseases. Most genes, phenotypes, and diseases are only seen in a single patient. And so we introduced Shepherd. Shepherd is designed such that it can integrate throughout the diagnostic process. After a clinical workup, which produces a list of phenotypes or symptoms that the patient is experiencing, after sequencing analysis, which produces an initial list of candidate genes generated using variant level filtering techniques, and after case review, which filters that variant filtered gene list down to a much shorter list of expert curated candidate genes. Shepherd is trained in a two-step process, embedding biomedical knowledge and embedding rare disease patient information. On the left, Shepherd is learning existing knowledge about rare diseases, such as the symptoms associated with each disease, the genes that cause each disease, the functions of each gene, the biological pathways that a gene is involved in, etc. This is done using self-supervised link prediction, where we predict whether or not an edge exists between a pair of nodes, and if so, what kind. On the right, Shepherd learns to embed each patient's phenotypes and a candidate genes or diseases in the context of the broader rare disease knowledge graph. This allows Shepherd to generalize to unseen genes, phenotypes, and diseases. Let me tell you more about how step 2 works. In step 2, the input is the patient's set of phenotypes, and depending on the task, a set of either candidate genes, diseases, or patients. We overlay the patient phenotypes as a subgraph in the knowledge graph. The candidate genes or diseases are simply individual nodes in the knowledge graph. We leverage the pre-trained model to embed each candidate as well as the patient's phenotypes. The patient subgraph embedding is generated by performing an attention-weighted average of the phenotype embeddings. We train the model using a loss function that encourages patient embeddings to be close to the embeddings of their causal gene or disease or other patients with the same causal gene or disease, and far away from the embeddings of the incorrect genes or diseases or patients with different genes or diseases. These notions of similarity that are captured in the embedding space can be used to rank causal genes or similar patients or characterize novel diseases. As a result, Shepherd is able to perform multifaceted rare disease diagnosis, causal gene discovery, patients like me identification, and novel disease characterization. In the interest of time, I will just focus on Shepherd's ability to nominate candidate causal genes. For causal gene discovery, Shepherd is given a patient's phenotypes and candidate gene list. Again, there are two possible gene lists, one that is a result of sequencing analysis and one that is shortlisted by clinical experts during case review. Based on this information, Shepherd predicts which genes are most likely to explain the patient's presenting symptoms. The performance is measured by average recall at K, with the number of causal genes retrieved in the top K ranked genes on average. As you can see by this dark blue bar, about 40% of the time, Shepherd is able to rank the causal gene first. We compare Shepherd to several representative domain-specific and machine learning approaches, and we find that for all values of K, Shepherd performs significantly better than most other baselines. It is comparable only to Lyrical, which is a domain-specific model. Now, let's take a look at a case study on a real rare disease patient to examine Shepherd's ability to nominate the causal gene of a disease. This patient was admitted in 2016 and diagnosed in 2019 with a POLR3-related leukodystrophy. Leukodystrophies are a group of rare genetic disorders that affect the white matter of the brain. The patient has an atypical presentation of leukodystrophy. There are several features of our clinical presentation that are not classically associated with leukodystrophy. When we take a closer look at this patient's data overlaid on the knowledge graph, we realize that while some of the patient's phenotypes in magenta are connected to the causal gene in orange in the knowledge graph, there are many patient phenotypes that are several hops away. Only 28% of the patient's 46 phenotypes are directly connected to POLR3A, and 94% of the 205 phenotypes directly connected to the gene are not associated with the patient. Still, Shepherd is able to rank the causal gene first in both the expert curated and variant filtered gene lists. Altogether, I have shown you the promise of contextual learning algorithms like Pinnacle and Shepherd to enable us to investigate the roles of a protein across multiple cell type contexts, nominate therapeutic targets in a cell type specific manner, identify causal genes of a patient's rare or novel disease presentation, and characterize never before seen disease presentations. I hope that I've convinced you that contextual graph machine learning can realize the promise of precision medicine, or at the very least, push us in the right direction. If you would like to learn more about graph machine learning, here are some great resources. Thank you very much for your attention. Please feel free to reach out via email if you have any questions.